Okay, moving along and continuing our introduction here to genetics. We're going to, in this video lecture, we're going to talk a little bit about how DNA gets replicated. How do you copy a DNA molecule? That's very critical. If a cell is going to divide from one cell into two daughter cells, each of those daughter cells has got to have a complete genome, correct? You got to copy all of the DNA and make sure each daughter cell gets a full set of those genetic blueprints. Because DNA, again, those are your recipe books. You got to have those copied if a cell is going to be able to operate and wind up with all of the traits that it's supposed to have. So DNA replication is a critical process for survival of any kind of living thing. And we'll see as we get into Chapter 5, viruses have to replicate their genomes as well, but they tend to, some of them get off into their own territory a little bit. So we'll reserve talking about viruses until we get to Chapter 5. All right, so what is DNA replication? Is this process of duplicating a DNA molecule. And now I'm going to warn you guys, your textbook is going into more detail than I'm going to require for our class. So yes, you're going to read about some of that detail as you go through your Smart Book, Learn Smart reading assignment, but use your study guide, learning objectives to see what I'm expecting you to know for testing purposes. So you're going to get some exposure to more detail as you go through your reading and other exercises, but when it comes to testing time, I'm zeroing in on some key things as we go through this particular chapter. So just keep that in mind. All right, so when DNA replicates, deoxyribonucleotides are the building blocks. They're going to have to get linked together in a chain to build a DNA molecule. And there's an enzyme for that. So we talked about enzymes. Enzymes catalyze these chemical reactions. Hopefully you're thinking about this being an anabolic reaction. It's a synthesis reaction. And hopefully you're remembering as well that requires energy. And now where is that energy going to come from? So we've already learned about ATP. Remember, that's a special nucleotide, adenosine triphosphate. That's an RNA nucleotide. But there's energy stored in those phosphate bonds. And as we're going to see, the energy that's needed for DNA synthesis, these DNA building reactions, comes from a very similar source. You've got phosphates in these nucleotides as well. There's energy stored in those bonds. You break those bonds, and that releases energy that can be used to build DNA. So the enzyme that's going to catalyze this reaction is called DNA polymerase. That's a DNA building enzyme. All right, and here's an example. DNA polymerase. What does it use as its substrates? It's got an existing DNA strand here, and it's got an incoming triphosphate nucleotide. So this is going to be the next nucleotide in our DNA strand that's being built. And if we blow that up, if we assume a G is going to be our next base in this DNA molecule that we're building, GTP is going to be the incoming version of the nucleotide, guanosine triphosphate. And that's what that looks like. So this should be reminding you a little bit of ATP. And we see there's a squiggly line there and a squiggly line there. That's indicating to us energy that's stored there that can be released to help power this DNA building reaction. And that's what they're showing you right here. Here's your existing DNA strand, looking at just one rather than two strands. And there's a sugar on this T nucleotide that has an oxygen and a hydrogen hanging off of it. That's your 3' end. Remember we talked about the 3' end of a DNA strand. Here comes your next nucleotide, which is going to be a G. The phosphate, the first phosphate on this GTP, is going to get bonded to the oxygen here. Those two phosphates are going to get broken off. The release of that diphosphate, those two phosphates, gives off energy that's used for synthesis. And now your G nucleotide is bonded to this single DNA strand. Now, what does this look like on a double-stranded DNA molecule? It's a little more complicated than that picture. All right, so if you think about in orange here, you've got a double-stranded DNA molecule and it's opened up. So if you're going to replicate a double-stranded DNA molecule, we're going to have to peel apart those two original strands and do some copying. And we say that replication of DNA is semi-conservative. What does that mean? All right, so if we think about this, back in the, think back, decades ago, before anybody had any idea about how DNA replicated, the early geneticists and scientists who studied DNA, they didn't know. All right, so you've got a double-stranded DNA molecule, and we need two. We need two copies of this DNA molecule. So when we go from one double-stranded DNA molecule to two, are the duplicates composed of all new DNA? Does the original go away and you wind up with all new DNA? Or do you wind up with an old DNA molecule with two old strands of DNA and a new DNA molecule with two new ones? Or do you wind up with your two DNA molecules having one old strand and one new one, and this one over here having one old one and one new one? And it was actually that third option, and that's why it's called semi-conservative. When DNA replicates, one double-stranded DNA molecule going to two, in each of those copies, you have one old strand and one new strand. So one old strand is being conserved in each new DNA molecule, and that's called semi-conservative, half-conserved replication of DNA. All right, so what we're seeing here is we've got an orange double-stranded DNA molecule that is being replicated, and if you're going to do that, you got to peel it apart. And if you notice on here, we've got this enzyme, DNA polymerase. This is a highly simplified diagram compared to what you have in your textbook. And all right, now what's going to happen over here? Notice down here, DNA synthesis proceeds from the 5' end toward the 3' end on the growing new chains. Okay, so DNA synthesis always goes from 5' to 3'. All right, and so the DNA polymerase on here hopped on to the old DNA strand down here and is sliding along. If it sees an A right here, it's going to put in a T. If it sees a G right here, it's going to put in a C. If it sees a C right here, it's going to put in a G. And it's going to be able to just keep sliding along because it's going in the 5' to 3' direction. Now over here, we need to keep peeling apart this old DNA molecule if we're going to keep replicating it. And there's an enzyme called helicase, and that's its job. It's to peel apart. It's going to break the double-stranded helix. It's going to peel apart those two strands. Your textbook also mentions an enzyme that's very important as well called topoisomerase. I'm not going to get into that, but that's important for relaxing what we call supercoiling of DNA. Think about an old phone cord and how it has a twisty helix-like shape, and you know how those old phone cords will wad up and fold up on themselves because of all that tension that's in there. That's a good way to think about supercoiling of DNA. It's all the double helix of DNA wads up and folds back on itself, and that helps compact the DNA down inside the cell. So all of that tension and folding has to be relaxed as well if you're going to copy a DNA molecule, and that's where the enzyme called topoisomerase comes in. I just want to mention that because you guys are going to be reading about that in your book. Notice what this is, primer, right in here in red. And again, this is going to get into a little bit more detail than we're going to cover in this class, but in your textbook, it will mention to you that this DNA polymerase can't just hop on anywhere on an existing strand of DNA and start replicating. It needs a primer. That's a little short stretch of, I know this doesn't make much sense, RNA. There's a little stretch of AGCU RNA right in here that is complementary to our old DNA molecule right in there. And there's another enzyme that makes that called primase. Then your DNA polymerase can recognize the end of that primer, hops on, and starts adding DNA nucleotides to build our new DNA strand, shown there in green. And then eventually the primer, because it's RNA, gets removed and it gets replaced with a little stretch of DNA in there. And again, those details are located in your textbook. I'm not really requiring you to know that, so but I just wanted to point it out since it's here on the diagram. All right. Remember how we talked about on DNA molecules, the two strands are always anti-parallel. So 5' to 3' on this one, 5' down here to 3' on this one. Okay. Now what's going on over here? Why do we have a little stretch of green DNA and there's a gap? And another little stretch of green DNA, and there's going to be a gap. And now what's going on over there? So your DNA synthesis always has to go from the 5' to the 3'. So I want you to imagine that original orange double-stranded DNA molecule being peeled apart, and this old strand goes 5' to 3' this way, correct? See that there on the diagram. That means the new DNA strands over here are going to have to go 5' to 3' in that direction. Your DNA polymerase can hop onto a primer right there and start building the new DNA strand. Okay? And then as the old DNA gets peeled more apart by helicase, your DNA polymerase enzyme is going to be able to hop on over here and make another piece of the new strand. And then as this keeps getting peeled apart, it's going to hop on over here and build another little short piece of DNA. So in a sense, it's almost like the DNA polymerase has to do this replicating or copying backwards on the other DNA strand. And those little pieces of DNA that are built are called Okazaki fragments for the Japanese scientists who originally discovered these little pieces of DNA that occurred in DNA replication. Now, eventually, those DNA pieces get sealed back together. There are gaps there that get bonded together so that you have a continuous strand of DNA. Okay. Go away. Now what's going on? Here's another highly simplified diagram of a replicating double-stranded DNA molecule. And in dark blue, those are the two old strands of DNA. They're being peeled apart. And in light blue, you see the new DNA strands being synthesized. This one's more simplified, so they're not showing you these little Okazaki fragments like you have on that particular diagram. But all that DNA polymerase enzyme is doing, it's working through complementary base pairing. A's and T's are complementary, G's and C's are complementary. And what that means is when it sees a C on the old strand of DNA, that's going to tell it to put in a G on the new one. A C over here, again, put in a G on the new one. T over here, put in an A on the new one. Those are called complementary base pairs. And that's how the DNA polymerase knows what goes over on the new strand that it's building. So this is how DNA replication works. How do you wind up with a faithful duplicate, a faithfully replicated copy of the original? And it's because whenever you have an A on the old one, you're going to put in a T on the new one. When you have a T here, you're going to put in an A over here. So you're always going to wind up with a copy of what you originally started with. Semi-conservative, again, your DNA molecule has one old strand and one new one. Here's another concept. DNA replication proceeds bidirectionally in two directions. And so what that really means is you're starting off with a circular DNA molecule, and all this business you see over here where DNA polymerase enzymes are making a new strand here, and over here they're doing the little Okazaki fragments to build the other new strand. That is going on in two directions. It starts at a place on a circular DNA molecule called the origin. So we got a circular DNA molecule here, double-stranded. There is a specific place on that DNA molecule called the origin where DNA replication is going to begin. and then the original strands of DNA are going to get peeled apart in both directions. They're going to get peeled apart that way and that way, and replication is going to proceed all the way around that circle, and eventually you wind up with a configuration that looks like this, where you've got two copies of a circular DNA molecule in this case, and they've got to be disengaged from each other, so they have to be nicked to make that happen, so that the two circles will be peeled apart. If not, they'll stay stuck together like that. So you got to nick the backbone so that they can be pulled apart. So that's called DNA replication. The point where you finish DNA replication is called the termination point. So DNA replication starts at a place called the origin and it ends at a place called the termination. What is an origin? There's information in the sequence of these A's and G's and C's and T's. So an origin is just a sequence of A's and G's and C's and T's that attract in the DNA polymerase and the other enzymes like helicase that are needed to replicate the DNA. DNA replication doesn't just start randomly on a piece of DNA. It has to, all that machinery has to home in on a specific location, and it can identify that location based on the AGCT letter code that exists there. All right, another feature of DNA polymerase enzymes that do this copying of DNA. So, here's an old DNA molecule, original, peeled apart. Here's our new strands that are being built by DNA polymerase. So every once in a while, when you have a T on the old strand, you're supposed to put in an A on the new one. What happens if the DNA polymerase accidentally puts in a G or a C? That is an error. And another term for that error is mutation, which we'll be talking a little bit more about later in this chapter as well. When you hear about a mutant or a mutation, that is a change in the DNA sequence that's generally caused by some sort of error. So DNA polymerase enzymes do make errors on occasion. How frequently? Some sources will say about one error for every 100,000 base pairs being duplicated. Your textbook says it's more like 1 out of 10 to the 8th, which is 100 million, or 10 to the 9th, which is 1 billion. However, in the finished product, when you take a DNA molecule and you completely replicate it, that finished product, you would typically find one error per 10 billion base pairs. So out of 10 billion base pairs, you might find one instance where there's a G paired with an A or a T instead of a C. And that's a mutation. That's a type of mutation. So what does this tell us? The DNA polymerase actually makes, maybe as frequently as 1 out of every 100,000 base pairs, a mistake. But in the finished product, it's about 1 out of 10 billion base pairs. So that tells us there's a proofreading mechanism that goes on. So there are DNA polymerase enzymes. There's more than one type of DNA polymerase enzyme that exists in cells, and I'm not going to hold you accountable for knowing the details about those different DNA polymerases, but just know that there are proofreading DNA polymerases that exist in cells that can remove mismatched bases. So if it sees an A and a G, they're not really going to be sharing hydrogen. Remember, A's and T's and G's and C's share hydrogen atoms with each other. That's hydrogen bonding. So if A is paired with a G by mistake, they're not sharing any hydrogens with each other. So they're not really going to be paired very well. And you're going to almost kind of have like a little bubble sticking out on your DNA molecule because of that. And so that mismatched base pair or pairs will get cut out. And then there's a DNA polymerase enzyme that comes in and fills in that gap. And that's called DNA proofreading. So usually these errors are corrected, but not always. And we get into mutations, we'll talk about what's the consequence if the proofreading doesn't work out. And when we get to viruses in Chapter 5, I want you to keep this in mind as well. Some viruses are good at proofreading their DNA or RNA as it's copied, and some are not. Some are very error prone, and there are lots of changes to the to their genomes, which ultimately lead to changes in their traits. And so just keep that in mind, because I'll bring that up again when we get to Chapter 5 on viruses. All right, so that's wrapping up this first section of Chapter 8. And in the next section, we're going to start talking about, well, how do we go from a stretch of A's and G's and C's and T's on DNA to building a protein that's responsible for a trait? And that's going to get into a discussion of transcription and translation, which are two processes that take place in cells to make that happen, this flow of genetic information from DNA to RNA to protein, which ultimately leads to a trait. So that'll be the topic we'll get started with in the fourth video lecture.